Okay, I want to thank the organizers for giving me the opportunity to present some of the recent work that I've been doing since I've moved from Brown University to Denmark and then after that to Padova. And that's why you will see that my affiliation is the University of Padova, but the work that I'm going to present, mostly from my students, they are all in Denmark at the moment. Okay, so there are nowadays we can measure somatic mutations in many cancer patients, and there are different large studies that have been conducted, for example, by the Cancer Genome Atlas and the International Cancer Genome Consortium. And what these studies have revealed is that in a typical tumor there are between hundreds and thousands or even more mutations, while there are probably a handful of driver mutations that are the mutations that are related to the disease, or maybe they could be related to clinical parameters in the patient. So one of the goals in analyzing this data is to identify driver mutations that are the ones that are important for cancer. So many approaches are based on single gene tests, in which one can assign essentially a score to a gene and then compare that score with the distribution for the score according to an appropriate null model, and then identifying genes that in isolation are significant. But I don't need to say to this audience that, of course, looking at genes in isolation is not the best thing we can do. We should consider genes in the context of networks. In particular, because it is thought that cancer is a disease of pathways. So it is a disease where mutations, driver mutations, actually hit a set of genes that are interacting, and by interacting perform certain functions in the cell. So our goal is to identify this set of genes that are significantly mutated or are associated with the disease. And as we have heard in the previous talk, what one can do is to start from known pathways, known set of genes associated with a certain function, and then one can easily generalize the score for the single genes to the set, and maybe look at these sets and try to understand if they are significantly associated with the disease. As we've heard in the previous talk, of course, this has problems, including the fact that we don't know all these processes. This approach also looks at these pathways in isolation without considering that they're actually interacting, and sometimes only a part of a set of gene, annotated set of gene, may be important for cancer. So the goal would be then to find this mutated set of genes without restricting to known pathways. And the main problem here computationally is that there are then many candidate sets. So if we look at all sets of genes, even of cardinality up to 6, we have more than 10 to the 22 such sets. So what I'm going to present today are two methods that I've developed with my students on leveraging on the information of large protein-protein interaction network and combining them with mutations to identify important subnetworks for cancer. So the first approach I'm going to present is a method to find subnetwork associated with survival for cancer patients. And it is a paper that will be presented in RECOMB next week. And the second approach is a more recent work on finding subnetwork mutated in a large fraction of patients that is not published yet. Okay, before proceeding, let me know that I'm going to talk about graph algorithms. And we can think about these graph algorithms as working directly on a protein-protein interaction network or on a graph that we can derive from a heat diffusion process that we deemed an influence graph in a paper in 2010 at RECOMB. And these graphs can be derived, as have been described, for example, yesterday by Linor, or with other diffusion approaches on the graph. And the main reason we want to do that is that the degree of the node in a network actually gives us some information about the confidence that we have in associating two mutations in a graph. So for example, if I have two mutations on two genes that are connected through a path of length 2, but that goes through a gene with a low degree, then I can have a certain confidence that these two mutations may be associated, while if instead I still have two genes mutated and they are connected through a path of length 2, but this path goes through a high degree node, my confidence is reduced that these two mutations are associated. And so by using heat diffusion processes, one can try to correct for these large degree nodes. And one way to do this, just at a very high level, is, for example, to build an influence graph in which we are measuring for each gene in the original graph an association with the other gene in the network. And we can do this for all possible genes, So at the end, what we derive is a graph that describes for each pair of genes some measure of similarity or distance if we want to look at the inverse of it. And I think there will be a talk later this morning that will describe this more in detail. OK, so now let's say that we start from our influence graph. And we want to find subnetworks that are associated with survival. So what's the data that we have? For many patients, we have measured mutations in all the genes in these patients. And we have time and sensoring information. Time is the survival time, while sensoring is a binary variable that tells us if the observed time is the exact time of the event, in which case we have a 1 for the sensory information, or if instead this is only a lower bound to the survival time of the patient, in which case we have a 0. So now let's assume that we already have a subnetwork. So for example, a subnetwork of 2 genes corresponding to these first 2 genes in this figure. Then we can measure the association of survival for the subnetwork by splitting the patients into 2 groups. The patients that do not have any mutation in these 2 genes, and the patients that have at least one of the 2 genes mutated. And we can derive then the survival, an estimate of the survival distribution for the 2 groups. For example, the red is the survival distribution for this group of patients, and the blue is the survival distribution for this group of patients. And by using known tools from survival analysis, like the log-rank test, we can obtain a p-value that is a measure of the association of the significance of the difference in, between the survival of these 2 populations. OK, so let me say something more about the log-rank test. So the log-rank test is a non-parametric test that is based not on the exact survival time of the patients, doesn't look at the time of survival of the patients, but we only consider the order in which we observe the event for the patients. So we can think that when we are performing the log-rank test for a particular subnetwork S, as we have described before, we are just assigning a group label to all the patients. So we're dividing the patients in a group 0 or in a group 1, according to the mutation status for that patient in that subnetwork. So the log-rank test computes a statistic that is a function of the group label and the sensoring information. And so what we have is that we have a weight for each subnetwork that depends in theory from the group label and the sensoring information. But since the sensoring is fixed in our data set, what we really have from the log-rank test is simply a weight for the subnetwork at n. OK. And what the log-rank test does then is give us the probability of observing a weight, a statistic, at least as extreme as the one that we have in our data under the hypothesis of no difference in survival between the 2 groups of patients. OK, so now what we can do then is use the log-rank statistic, the weight given by the log-rank, to define formally a computational problem that we will use to identify subnetworks associated with survival. So given our mutation data, the survival information, and the large gene interaction network, and the value k, we want to find a subnetwork of k genes connected that maximizes the log-rank statistic, so the weight given by the log-rank. So in the paper that is going to be presented at Recomb next week, we prove that actually this problem is NPR by reduction from the set cover problem. And then we present an algorithm called NOMAS to identify these networks of mutations associated with survival. So NOMAS is based on a color coding algorithm that is a technique that has been used a lot in bioinformatics. And what it does, it identifies the subnetworks of mutations associated with survival. And now, since we have optimized to find subnetworks such that their mutations have a high association with survival, we need to test that the association for the group that we observe is actually not due to chance, just to having picked a random set of genes in the network. So what we do, we also perform a permutation test in which we fix the mutation, the survival time of the patients, and we randomly permute the identity of the genes in the network. OK. So as I said, color coding, it has been used a lot in bioinformatics, and these are just some of the papers that use color coding for bioinformatic problems. And let me give you a short introduction to how color coding works. So if we're looking for a subnetwork of size k equal to 3, and this is our graph, we now want to find a subnetwork that maximizes certain objective function. What we can do is we can random color each vertex with one of k random colors, obtaining a graph that has random colors on the vertices. And now, once we have fixed the coloring of the graph, we can find the highest scoring colorful subgraph that is a connected subgraph that contains all k colors pretty efficiently using a dynamic programming approach. And this is pretty efficient when k is small. In particular, when k is at most of the order of the logarithm of the number of vertices in the graph. Okay, so how does this algorithm, this dynamic programming approach work? Well, essentially, we build a table in which we have genes on the rows and the different color sets, the different possible subsets of colors that we can have in the network. And for each gene, we are going to identify the subnetwork containing vertices with those colors, and exactly that number of vertices, that give us the highest weight. So for example, if I'm considering this entry, let's say that this row corresponds to this specific gene. Now I'm trying to identify a subnetwork of two vertices that contain only vertex that is one blue and the other one red. Since I'm in this vertex, I already have the blue gene, and I have now to consider connected vertices to this gene that are red. So I have two candidates, this subgraph and this other subgraph. I can compute the score for each of them, and then I will record into this entry the highest scoring solution. And then when I will try to build a subnetwork that contains this vertex and will contain three different colors, I will try to merge this subnetwork with this other one. Okay, so if the highest scoring subnetwork for our problem is colorful in our graph, we will identify it using this approach. However, since we have used a random color, it may be that the best scoring subnetwork is not colorful. So what we can do is repeat this approach many times, and at the end, we can report the highest scoring subgraph that we identify. And one can actually prove that the highest scoring subnetwork will be colorful at least once if one repeats this process enough times. Okay, so one can use this approach or a variation of this approach for our problem, in which the score that we are optimizing is the log rank statistic. But what we have is that if one looks carefully at it, the approach for color coding works when the score is additive. So when the score for a set of genes is the sum of the score of the single genes. And the main point is that for the log rank statistic, the score that we have is not additive. So actually, it may be that even if the optimal solution is colorful in our graph, we may not be able to identify it with this approach in the worst case. However, what we prove in the paper is that under a model that is, we believe is kind of reasonable for mutations and survival, if we have enough samples using the approach based on color coding, we will be able to identify the optimal solution with guarantees essentially similar to what we have for approaches in which the score is additive. So we don't lose anything when the data comes from a certain distribution by using the color coding approach. Okay, so let me now show you just some results of the application of NOMAS on cancer data. This is essentially ongoing work, in particular for the results that I will show for one of the subnetworks that I will show. So the first thing we have done is we have compared our approach NOMAS with two other algorithms, or two types of algorithms that have been proposed before for the same problem. One is a greedy algorithm, in which we build a solution starting from a vertex and adding other neighbors by trying to maximize the increase in the solution. And another one is by using an additive score. So the score for a gene, for a subnetwork, will then be just the sum of the p-values for the single genes, for example. So by
Fabio, can you expand this last point? I thought you said that you proved that you can use the normal additive color coding to find the highest scoring network. So
Yes.
How do you compare to an additive? Something I missed.
So there are some approaches in the literature that to identify, you can assign to each gene a single score, like the p-value for that gene or the log-rank statistic just for that gene. And now you try to find subnetwork that maximizes the sum of these scores. The score for that subnetwork, though, is different from the score that you get for the log rank in our model. Because now you are looking at each gene individually, and it's different from taking just the entire subnetwork and partitioning the 2 patients for that one.
That I understand, but in your color coding approach, you use an additive score.
No, it's not additive. Our score is not additive. That's the main point. Thanks for pointing out that it was clear. So our score is not additive. And what we prove...
Your theorem says that even if you use an additive score, you're guaranteed to find the maximal subnetwork.
No. What he's saying, or I hope I have written what I wanted to say, is that if the mutation, the optimal solution are independent of survival, that's our model, and you give me enough patients, this M, then NOMAS, our approach based on the color coding, will identify the optimal solution according to our score that is not additive.
But still it will implement an additive color coding approach. So this NOMAS is
now when you compute,
additive scoring.
okay, now I see when I compute the score for a subnetwork now, I don't add over the genes, I compute the score for that subnetwork. And then when I join 2 subnetworks, what I will do is I will recompute the score for that subnetwork. I don't sum the score for the 2 subnetworks.
So you're saying that using NOMAS, you first find the subnetwork using your additive scoring function, and then you evaluate the network.
Yes.
Using the non-additive, and it's still going to be the best.
Yes. When we try to find these candidates, we use essentially an additive version, but then for each additive, we are recomputing the new score. Okay. Okay, so what we have when we compare NOMAS with these other approaches, different variants of the greedy algorithms that there could be, or by using an approximation essentially of the score based on the sum of the weights on the single genes, what we see is that NOMAS returns subnetworks that have higher correlation with survival. So they have higher association with survival than the other approaches. Sometimes the results, the subnetworks that are returned are similar, but sometimes they are quite different. In particular, using the additive score returns sometimes subnetworks that are not as highly associated with survival as it is instead by using the greedy algorithm or NOMAS. Okay, so now let me show you one specific subnetwork that we identify using data from glioblastoma multiforme in TCGA, in which we have 266 samples, and here we use somatic simple nucleotide variants and copy number aberrations. So what we identify is this subnetwork of 8 genes, in which part of the subnetworks correspond to the PI3K AKT signaling pathway, and part of the other subnetwork contains genes in the P53 RB1 pathway. So there is some network information and correlation between these genes. And these are the 2 survival distributions for the patients with no mutation in the set of genes and for the patients with the mutation in the set of genes. So this difference is significant, so if one uses the log-rank test, the p-value is quite small. And in addition, if one does this permutation test on the network, it obtains that it is unlikely to see a set of 8 genes in the network that show such association with survival. Okay, and let me point out that none of the genes in this group is associated with survival when considered in isolation. So you will not identify this set if you look even for single genes and try to add new genes to it. Okay, so now I'm going to move to the second problem. It is the problem of finding subnetworks mutated in a large fraction of patients. So I'm presenting this problem at the end, but actually, historically, I should have presented it before, because this is a problem that we formulated in exactly the same way I'm going to use here in the paper that was a Recon 2010 in collaboration with Eli Upfal and with Ben Raphael, in which we had 2 approaches that we suggested to identify set of genes that were mutated in many patients. We call the first one a combinatorial model. That is what I'm going to talk about today. And the second one is what we call the enhanced influence model. The enhanced influence model should have really been called HATNet, because that's what was later called HATNet, even if the name doesn't appear in that paper. And this method is what then has evolved in a better version that is now called HATNet 2, and has been published recently, both based on the influence graph that I've described before. Okay, so what we will do in the combinatorial model is identify subnetworks that are mutated in many patients. I'm going to define formally the problem in the next slide. And then we will use a permutation test to assess the significance. So just generate a certain number of permuted data sets and look in the fraction of data sets in which we identify solution at least as good as the one we see in real data. Okay, so in the combinatorial model, formally the problem is the following. We take the influence graph, we combine it with mutations in the sense that what we want to do now is find, for a given value k, the subnetwork, the connected subgraph, it is the influence graph, that is mutated in the maximum number of samples. So this is the problem, and we called it in the paper the connected maximum coverage problem. That is essentially a graph, a version of the maximum coverage problem in which you pick k elements trying to cover as many sets as possible. That is a non-NPR problem. And this is the same problem essentially just with graph constraints on the solutions. This is related also to a problem from Ulitsky, Karp, and Shamir, in which they had a different formulation, which they wanted to cover to find the smallest connected subnetwork that covers, let's say, almost all samples at least k times. And this should be true for a large fraction of the patients. Okay, so what we proved in the paper in 2010 is that this is an NPR problem. In general, even it is an NPR problem even for simple topologies of the graph. And we proposed an approximation algorithm to solve this problem. And the approximation algorithm is essentially a generalization of a simple greedy algorithm in which we build a solution starting from each vertex, and then we will pick the best solution at the end. And for a given vertex, we build the solution starting from the gene, looking at paths that go from that gene to the other ones, path of a certain length. And then we will add to the current solution the path that give us the maximum increase in score, but weighted or averaged over the number of new nodes that are added in the solution. Okay, so this was an interesting algorithm, at least, and I found out recently that has been used in other scenarios, not that have nothing to do with the computational biology. So this has been used in problems in wireless networks, where you want to cover many users by placing routers, and they should be connected so that you minimize some other function that I'm not sure what it is. And what they showed that experimentally, even for these other problems, it worked well. So it worked as well as the approach they were suggesting, at least for the cases that they could solve. Okay, so the algorithm worked well in finding these high-scoring solutions, but it's not an exact algorithm. And while talking with the collaborator at University of Southern Denmark, he said that he thinks that an ILP solution may be faster and still give of course, the best solution. And so while discussing that, we started working together, and so we came up with an ILP formulation for this maximum connected coverage problem. So just going quickly through this ILP, what we want to do is to cover the maximum number of patients, collecting k subnets, k genes that we cover the patient. We cover a patient only if there is at least one of the genes that we pick that is mutated in that patient. And then we have to make sure that we have a connectivity for the genes that we pick. And here the connectivity is defined by this set of constraints that, without giving too many details, require that for each pair of genes I and j in the solution, if we look at node separators, so a set of vertices that, if removed, will disconnect these two genes, at least one of them should be in the solution. And then, based on this, one can use a simple branch and cut strategy. Okay. So we have implemented it, and then we have compared the runtime for the ILP with the runtime for the approximation algorithm and the greedy algorithm for different data sets from TCGA. And so what we found is that actually this ILP is quite fast. So it's much faster than the approximation algorithm, because the approximation algorithm had to compute many paths. And it is also comparable in running time with the greedy approach. So this ILP that finds better solutions on the data sets that we identify, better in terms of the coverage, so the coverage that we identify is better than the coverage we see with the greedy approach or with the approximation algorithm. And still, the runtime is comparable. Sometimes it's faster, actually, than the greedy algorithm. And so it will allow us also to run permutation tests efficiently. So as I said, this is work in progress. So I don't have results to show for the biological networks that we identify. We're evaluating them at the moment. But what we see is that if one then uses the approximation algorithm to identify subnetworks, evaluate their statistical significance, or uses the ILP to identify subnetworks and then assess their statistical significance, the results can vary quite a bit. So in particular, what we have is that for some of the datasets from TCGA, using the ILP, we will identify some significant signal in the data, while using the approximation algorithm, actually, we don't identify such significant solutions. OK, so with this, I'm just going to acknowledge the people that have done the work that I presented. The first part of the talk on NOMAS is the work of Tom Jansen, a student of mine, University of Southern Denmark. And the second part of the talk is ongoing work with Anna Bommersbach, that is a student that I'm co-supervising with Marco Chiarandini, my colleague at University of Southern Denmark. So thank you, and if you have questions, I would be happy to answer. I guess actually it was. Sorry about.
On your left line, it says statistical significance of a subnetwork. What do you mean?
So what we do once we find the subnetwork, it's a subgraph that covers a certain number of patients. Then we ask what's the probability that if we permute the identity of the genes in the network at random, we find the subnetwork with the same number of genes that covers at least that many patients.
It's not functional relevant.
No, structural, yes. It's significance in terms of the statistic that we are observing. That is the coverage of patients, yes.
So I have a question about the assumption that you use of independence of the mutations that are not in the optimal subgraph for the survival, but those other mutations are independent of survival. We know examples that's not true. Do you have a way of testing that so that you know the context in which cancer the model can be applied and in which it would not be appropriate?
By saying that we have example where that is not true, you like that there are
it would not be true, as we know, if you have one subtype that have apobec activities and other subtypes that don't. Because apobec activity will generate random mutation, and we know that apobec activity is associated with bad survival prognosis, at least at breast cancer. So then, you get this random passenger, you assume that they are independent of survival, when in fact they are. So do you have any way of testing it that's indeed the case before you apply the model?
We haven't tried that. No, we didn't try to correct for that, actually. That's a great idea, and I think it's a great direction for future work. Ideally, if one has a way to assess that, then you can split the patients into the two groups and see what happens. But that's a great point. Yeah.
Have you looked at whether things that have a high allelic fraction, so these are things that you would think would probably be drivers because they've sort of come to dominate the cancer cells, do you see those showing up in the networks that are survival associated?
We didn't check that also because we don't. There is some data that one can use to assess that, but for the many patients that we have, the coverage is not as high if you want to do a very refined analysis. It depends on what you want to do exactly, but yeah, that's a great point, and we didn't check for that.
I think TCGA, since this is TCGA, I'm pretty sure they'll give you a allelic fraction for the mutation.
Yes, you get the allelic fraction, but I guess the coverage usually is like 30x, so it depends what you are really looking for. So if you want to distinguish different things, I'm not sure the 30x is enough. Then if you just want to say if there is a correlation between the different, what we see, if it is just highly, if it has high variant allelic fractions, then yes. But yeah, we didn't check. That's a great idea, actually.
So in your second part, you said your integer linear programming approach is quicker than 3D algorithm.
Yeah.
Usually, I mean, I'm just curious that usually 3D algorithm should be much more efficient. I mean, much quicker than integer linear programming. Right? But I'm just curious what trick you use in your.
I don't think there is a trick, honestly. When I was speaking with my colleague, I was saying exactly the same thing. So that, the ILPU is going to be slow. Here, the trick, if you want, is that you can, again, it is only for these instances that it is fast. There may be, of course, instances in which it's not going to be as fast as the greedy. The trick is that you can generate these constraints very efficiently for your branch and cut. So it's very easy to find the node separator that is violated once you have a solution is not connected.
The data size?
Data size, I guess. So these are the number of patients. And I guess we are looking at subgraph that has 15,000 vertices. And that's the number of edges.
So I wonder if the permutation test is enough, because if you have so you're just renaming nodes in the network randomly. But if hub nodes, for example, tend to have more mutations, then you are not capturing.
For the second part, I guess?
Yeah.
Yes, if there is a high correlation between the degree of the node and the number of mutations, then yes, there would be there could be such an effect that we are not capturing. I have to say that for some other analysis that we did, we checked the correlation, and there is some, but it's not so strong that it would completely, let's say, make the problem, that it will make the solution not significant. So, yeah.
So for the two parts of your talk, you seem to be focusing on different things. The first one is distinguishing people who survive longer than the ones that don't. The second one is trying to cover as many patients as possible. So do you find similar subnetworks or not at all?
Not at all, actually. No, I shouldn't say not at all. So for the GBM one that we showed, actually, it's very similar what you would find, what you find with the two methods. But the ones that we have other cases in which the subnetwork associated with survival is not the one that is covering the most patients, or in general, there is no relation between it.
Which one is better?
I think they answer different questions. So one is looking for the survival, the association of mutations with survival, and the other ones instead is trying to explain as many patients as you can, essentially. Trying to find mutations that explain as many patients as you can, saying that those are going to be the driver mutations for cancer.
But if they are both finding driver mutations, shouldn't they converge?
I guess they are using different signals, and there might be driver mutations that are not associated with survival. Like a simple trivial case is like a mutation in a gene that is mutated in all the patients with that cancer type. There's no association with survival just because everybody has it, so you cannot even think about not having it. That's a trivial one, but there are many cases of driver mutations that actually don't have association with survival. So.
One more question.
Okay.
So I guess if you wanted to get at the permutation test where you break the association with survival but preserve the network, you could shuffle people's survival status but keep the network the same. I assume you haven't tried that, but...
Yeah, we have tried a many different permutation tests. So one is this one, just permute the identity of the genes. That is the one that actually I like the most, because it's saying, with our method we are finding a set of genes, and we want to see if the set makes sense. So, if it is the connection of these genes that makes sense or not. And then we tried permute the survival of patients, keep the same mutation status, and just permute the survival of the patient. We tried that, and they are still significant. We've also tried to just assign the mutation in each gene independently and keep the rest the same, and you get still significant stuff. So, yeah, I think is a good way to do it, but I like this test more, I have to say.
Yeah, I think that one would have addressed the earlier question.
But the earlier one, if I think, wasn't about survival, right? The first one was on covering patients. So in that case, if you cover patients, we are not looking at the survival stuff. So.
Thanks again.
Thank you.